Welcome, everyone. Thank you for joining us at Navigating Spatial Biology. Take that next step with Leica Biosystems. Our next presentation for today is Implementation of Fully Automated Bond RX Protocol for NanoString's GeoMX Assay, presented by Carlee Hemphill. My name is Marie, and I will be your moderator for today's event. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want during the presentation. To ask a question, click on the Q&A box located on the bottom of your screen, type your questions, and hit Submit. The speaker will answer your questions following the presentation. If you have trouble seeing or hearing the presentation, please click on the Q&A box to let us know that you are experiencing a problem. Please join me in welcoming Carlee. For a complete biography on our speaker, visit the Biography tab on the left. Carlee, over to you.
Hello, everyone. Good afternoon. My name is Carlee Hemphill, and I'm a lead scientist here at the LabCorp Drug Development System. I want to thank the moderators for having me here to present today. I'm going to present on the implementation of the fully automated Bond RX protocol for the NanoString GeoMX Assay and what we've done so far here to make sure that it is working in the beta version before the commercial launch. Just for a quick GeoMX DSP technology overview for those unfamiliar, this is a relatively new technology that combines the ability to have a pathologist read along with an nCounter or NGS barcoding output. And this allows us to resolve and quantify either protein or mRNA expression in a spatially resolved manner. So the assay uses either antibodies or RNA probes, and these are coupled by NanoString to photocleavable oligonucleotide tags. And these tags are combined with up to 3 morphology markers plus a nuclear stain to FFPE mounted tissues. The oligonucleotide tags are then released from selected regions of interest based on the morphological markers, and they are released from the tissue by UV exposure and collected into a 96-well plate. These tags are then quantitated in a standard nCounter assay, or more recently, as we have implemented by a next-gen sequencing by Illumina, and then mapped back to the tissue location. So again, this allows us to have a spatially resolved high-plex digital profile of a certain type of analyte abundance without doing any type of degradation to the tissue or loss of tissue integrity. So the big 3 points of the NanoString GeoMX is the digital spatial profiling aspect. So first, the digital, as I mentioned, the multi-dimensional analytes are counted using nCounter or an NGS by Illumina. The spatial, this allows us to identify regions of interest based on either protein, RNA scope, or morphological markers. And this allows us to have a limited number, up to 4 fluorescent tagged antibodies or RNA scope probes that'll help guide the scientists to a region of interest that is interesting. And then finally, in the profiling, the counts are mapped back to the tissue location of the ROI selecting. And so there you have yielded a spatially resolved digital profile of the analyte abundance, again, either protein or RNA. So this is a typical workflow for the technology. Again, part 1 is staining either with those antibodies or with the RNA probes. Then we do the secondary stain with the morphology markers. So that, again, can either be fluorescently labeled antibodies or RNA scope fluorescently labeled probes. They are then scanned on the DSP instrument and the ROIs are selected. This can be done remotely if you are VPN'd or a remote desktop into the instrument, or it can be done on site. And so from there, once the ROIs are selected, the instrument initializes the UV cleaving and collects those into the 96-well plate. And from there, they're either read out on the nCounter for the RNA plex assays up to 84, or for the higher plex assays done on an NGS readout and then brought back into the DSP for that spatial resolution. So this is the timeline of our adoption here at LabCorp of the NanoString GeoMX Profiler. So we purchased the instrument in November of 2019. We had it installed at our companion diagnostics lab here in Raleigh, North Carolina. And then from December 2019 to February 2020, we did all of our standardized instrumentation, operational and performance qualifications, and then training with the NanoString scientists and like a specialist for both the Bond RX protocol, the protein IO panel, and the RNA IO panel. Again, this was a good 3 months of training to make sure we were familiar and comfortable with the system. And then beginning in March of 2020, we started our own internal projects, expanding our experience. There was a presentation done for the 2020 AACR that shows the implementation and how it correlates to different types of outputs in both multiplex fluorescence and the IO360 profiling. And as we all know, in April 2020, everything in the world changed. So our first collaborations with a lot of our pharma partners were put on hold as we were trying to onboard these, which gave us a lot of time to really sit down with the pharma partners, come in and make the corrections to the protocols and definitely a study design. So this time actually gave us a lot of time to reflect on how we can best design experiments, and then it certainly gave us an opportunity to test the remote capabilities of the DSP. So I could be sitting safely at home under quarantine, but I could still reach our NanoString DSP in the lab 20 minutes up the road and work with pathologists to find our regions of interest or design experiments. And then in May 2021 of this year, we brought on our first custom panel. So we had a client come in asking for specific mRNAs that were not in the custom or in the off-the-shelf IO panel. And they also wanted a morphology marker that had not been validated by NanoString. So this has been a big bulk of our work this year. In addition, we have onboarded the Illumina NGS workflow. So that has been a huge increase. We can now run the CTA, which is approximately 1,800 genes, or the whole transcriptome WTA, which is 18,000 genes. We have onboarded that here at LabCorp. And then most recently, we brought on the Leica team to upgrade our Bond RX with the fully automated protocol that they had developed with NanoString, and making sure that we can, for our workflow, going from a semi-automated staining to a fully automated run, and really reducing the bench time needed by the scientists. Okay, so on this slide, you can see the overview of NanoString's current available panels. First on the upper left is the protein assays nCounter readout. This is the immuno-IO core that I mentioned that we had done the full training on. So this has 18 different protein targets that are read out by the nCounter. In addition, there are 7 off-the-shelf add-on modules that range from an additional 5 to 10 antibodies of interest. Anything from apoptosis to MAPK signaling can be easily added on as a module. And also this allows for full customization. So any antibody of interest, as long as it can be labeled by the oligonucleotide tag and or be fluorescently labeled for a morphology, we can add these into that protein assay readout. On the upper right-hand corner is the RNA assays nCounter readout. So this is the one for those who are looking more for RNA analyte abundance. The IO core has 84 immune pathway targets, and you can also add up to 10 additional RNA probes. So these probes are designed in silico by NanoString and delivered to LabCorp as a set that just gets added onto that IO core, and it is also read out by the nCounter. And the two newer assays that have been developed are on the bottom. These are for the NGS readout, which is an Illumina-based sequencing. So there's the protein core, and there's an additional 10-plex module that can be added on. So this can do up to 100 protein targets and get an NGS readout from those. Again, The modules are flexible and allows for customization and really increases the throughput on the amount of, or increases the complexity of the protein assays that you can run. And then the big players over on the lower right are the RNA assays for NGS readout. So we have the Cancer Transcriptome Atlas, which, as I mentioned, was 1,800 genes, and then more recently, the Whole Transcriptome Atlas, which is available both in human and in mouse. So either one of those can be used for these types of studies, and that includes a little over 18,000 genes. So what does a scientist need to do in the lab based on the current workflow? So it's up to a 5-day turnaround time just for data. So on day one is a slide preparation, which is where we do all of the the baking, epitope retrieval, staining of the probes or antibodies, and then those do an overnight hybridization. On day two is where we do the morphology staining, and then we do the slide cleanup, and we do any type of stringent washes. We scan it, select ROIs, and then collect on the DSP. Those probes that come off in that 96-well plate are then hybridized with the nCounter or NGS tags, and they're hybridized overnight. So minimal hands-on time there. Day three, those analyzed are collected by the probes, either, as I mentioned, by nCounter or NGS, and put back onto the DSP. So we finally have that spatially resolved analyte abundance. And then day four is where the data interpretation really starts, where we can look at the QC of the run. We can do any type of normalization on the data. We can do background correction, any type of statistical analysis within the software. And then day five is really spent on data analysis, tying up those loose ends, and then report writing and getting the information back out to the client. So again, this is really impressive that from sample to multiplex data, we can do this in under a week, but we are limited by the amount of slides that can be put on the NanoString GeoMX, which is 4 at a time. So we really have a low throughput, even though it's only a 5-day turnaround. And so this part right here, this slide cleanup and the stringent washes and the scanning is where the big bottleneck is, and that is because it requires a lot of hands-on time for the scientists. So instead of starting a new run or starting a second set of scans, the scientist is spending up to 5 to 6 hours of stringent washes, doing the morphology markings, and then getting those loaded onto the DSP. So if we can fully automate this step, we can really increase workflow. So this is the previous for what I'm going to be referring to as the SemiAutomated Bond RX assay. So the day one and day two hands-on time is where we are losing a lot of critical space and where we're losing a lot of our throughput availability. So on day one, obviously we bake slides and prepare the Leica reagents. This takes up to an hour. Then there's a 3-hour on the Bond RX protocol that does the epitope retrieval and washing. We then offline prepare the hybridization mix and then manually pipette the probe mix onto the slides, apply the hybridizing, the hyb slip, and then put them into a hybridization chamber for 16 to 18 hours overnight. Day two, we have to come back, take them out of the hybridization chamber, and there's 90 minutes of stringent washing. So this really is a lot of hands-on quality time that the scientist is losing doing stringent washes that could be automated. There's an hour-long incubation of adding morphology markers on day two in the morning. So that is also lost time that could have been automated. And then again, after that, morphology markers are added, the slides are washed, and then loaded onto the DSP, which is an additional 30 minutes. So again, while it is semi-automated, it is still a heavy hands-on approach to the assay. And with one operator and run instrument, we were really limited to completing 4 slides a week. Then Leica and NanoString partnered up and came up with the fully automated RNA protocol on the Bond RX. So as you can see, the decrease in day one and day two hands-on time is very minimal. We still had to bake slides and prep reagents, but instead of just waiting for that 3-hour run and then doing RNA probe hybridization and or morphological markers by hand, it is a fully automated, approximately 22-hour run on the Bond RX. And then day 2, the slides come off. It is just a removal of the slides from the instrument and right onto the DSP. So at that point, we can actually prep reagents for a second run, get 4 more slides on, start that 22-hour run while we're doing ROI selection day 2 on the slides run before. So again, this really is a way that we can improve turnaround time, improve throughput. And over on the right, you can see that we're not, this is a picture of a 4-plex morphology panel, and this was utilized with the fully automated Bond RX protocol. So we're still getting great staining details, and we're not losing any quality in the data because of the fully automated run. So again, in summary, we do have an increase in workflow and turnaround, increase in workflow and a turnaround and a decrease in turnaround time. So we have significantly reduces the hands-on time for each slide. We have increased throughput, as many slides can be stained on the Bond RX and in one run. And this increases the throughput on the DSP as the operator is not tied to the bench lab work. We recently completed 16 slides in 1 week with 1 operator and 1 instrument. So as you can tell, a 4-fold increase in productivity and ability to turn these assays around becomes much more beneficial. So while we were very pleased with how the fully automated assay was running on the Bond RX, we wanted to make sure that we were getting the same quality data we were doing with the manual run. And while NanoString came to us and said, we're beta testing, rolling this out, would you be interested in partnering with us and seeing if we are getting the same amount of data qualitative for the specific assays that you guys are used to running? So we came up with the robustness testing experimentation where we were looking at background signal and does the background signal intensity change between the automated and the semi-automated run? Is the housekeeping concordance similar? So are we seeing the same housekeeping genes at the same frequency? And then finally, is the biological concordance upheld? So does the interpretable biological outcome differ between the assays? So here you can see 2 graphs. These are the negative probes. So this is basically what your background is going to be looking like. On the left is the semi-automated. So these are counts per different negative probe across 4 different slides, and with 12 ROIs per slide. And then over on the right, you can see the box plot for the fully automated run. While we are seeing less variability in the fully automated run, the more important part is we're actually seeing a decrease in overall amount of negative probes that are showing up. So we're getting a very large fold decrease in the amount of background probes, meaning that the automated assay is actually giving us cleaner data than we were getting with the semi-automated assay. And this makes sense. It being a fully automated run, the instrument is much more capable of doing reproducible washes, of doing more stringent washes, of making sure a 5-minute wash is exactly 5 minutes instead of 5 minutes and 2 seconds, making sure the staining time of the probes is exactly at 15 hours, where it can be a little more variable between 16, 17 hours, depending on when the scientist was in the lab and able to return to the experiment. So this was really comforting, knowing that we were getting a decrease in the amount of negative probes seen with the fully automated run. So this is actually an increase in the data analytics as far as we could see. And so here's the data comparison for the housekeeping genes. So on the bottom, you can see different housekeeping genes across different scan IDs. So these are the different scans that we had run. So this one we had six different samples that were run comparing the semi-automated to the fully automated. You can see the housekeeping genes, share a similar pattern. We're seeing the same trends, but we're also actually seeing an increase in the housekeeping genes raw count outputs. So on the fully automated on the run, we're seeing, housekeeping genes registering in the thousands, whereas with the semi-automated run, we were getting a much lower signal intensity. So we also found this to be a plus, that we are getting much more analyte readout from the same samples on known housekeeping genes because of this fully automated run. And I think the reasoning for this is exactly the same as we saw with the background. We're just having a more consistent and more reproducible assay environment that allows us to really increase the integrity of the data. So I think this was, we're seeing the same trends, so we don't think this is anything that is biologically insignificant. It still makes sense in the context of the story. It just makes for a better story because we're getting so many more counts and so much more data above the threshold for the housekeeping genes. So this was really exciting as well. And then lastly, we wanted to look at the data comparison for the biological concordance. So I just picked a small subset of the probes that are available. I kind of tried to pick ones that were known and everyone's familiar with. So we have, some of our CD3, CD4 markers, CTLA4, FOXP3, LAG3. Everyone who's familiar with the IO space is really interested in what we were seeing as far as the fully automated versus the semi-automated. So again, we can see the scale bars are a little skewed on the side just for graphical reasons to be able to fit them on the same. But if you do look at the scale bars, you can see that we are getting the increase in counts for the fully automated run versus the semi-automated run. But we're seeing the same trends. We're seeing that CD86 is still much higher. So that biological concordance is preserved, and the PD1 is still higher than the rest of them. So we're seeing that same biological concordance, but we're seeing it at a much better scale. So we're getting higher counts, higher readouts, and therefore better signal intensity with the fully automated run. So in conclusion, we really appreciated the ability of Leica and NanoString to work together to allow us to onboard this fully automated protocol, producing not only an increase in our ability to get more slides done in a week, but also increase the quality of the data coming off of our instrument because of the lack of variability between runs. So thank you so much for your time, and I welcome any questions.
Thank you, Carlee, for that informative presentation. Just a quick reminder to our audience on how to submit questions. Simply type them into the Q&A box and click on the send button. We will answer as many questions as we have time for. Our first question is, how can spatial profiling workflows help in gathering multiplex data?
Thank you for that question. So the spatial profiling aspect really helps us get higher dimensional data. So from traditional methods such as IHC or MIF, we are limited in the number of markers that we can explore. But with the spatialized data, we can still get that spatial context, but also certainly get a lot more data points from the ROIs.
Great. Next question. Are there any similarities between spatial profiling and more traditional multiplex methods?
There's some similarities. You can see from the presentation the multiplex IF staining, so that basically builds on the traditional methods of the more traditional analysis. But the ability of the sampling to look at several different phenotypes and to look at several different signaling pathways gives it an edge on the current traditional assays that are available.
So yeah, I was going to follow up with any differences to consider.
Differences, really just the workflow, the ability to get large amounts of multiplex data from the analysis compared to just a visual representation. You can certainly different types of analysis downstream of multiplex IF, but the digital spatial profiler really allows you to get a more comprehensive view and larger amounts of data.
Great. Next question. How do the morphological markers help guide the selection process for ROIs?
So the morphological markers are really helpful. First, they're customizable, so any type of target that you're looking for, as long as you can fluorescently label that antibody, you can use it as a morphological marker. So this really helps us to get customized work done, and especially if there's a specific drug target or a specific cell type that we're looking for, information around that cell. It helps us really hone in and go, make sure we are getting the correct ROIs and getting the answers for the questions that are biologically relevant.
Thank you. Next question. Can we use it for mitochondria function analysis?
Let's see. I personally have not used it for any type of mitochondrial function, but certainly the gene list exists, and so I don't see any hindrances to why it would not work. You can't get down into the single cell phenotyping, but if there's specific pathways you're looking for in a general area, that you could definitely utilize the DSP for that type of information.
Great, thank you. And it looks like we have time for one more question. What are the most common areas of research that your clients are using DSP in your lab, outside and within oncology?
So certainly oncology is our biggest client interest, and immuno-oncology is probably 90% of that work. And so, but we're also seeing an increase in the neuro panels that are available. So a lot of interest in Alzheimer's, any type of disease aging process, those have started to come in. So that is, the biggest areas that we see. And then, so I think another big part of DSP is the availability of the customization. So we have a lot of clients that come in and have a very specific drug target. And so if there's an antibody to, that drug target, we can see if that's localized within the tumor, see if that, what is going on in that area where the drug target is either entering or unable to enter into the tumor for efficacy.
Great, thank you. It looks like we do have some time for another question. When is the whole transcriptome assay being used by your clients?
I'm assuming this is a when in not a timeline sense, but a when as in an experimental sense. So I'll answer it that way. It is currently already being used, so I guess that's the answer to the timeline. When people are using it and engaging with it is really for early discovery phases. So if there's a specific mechanism of action study that is of relevance, those are very popular with the whole transcriptome atlas because you can really go fishing for large amounts of and get a lot of amounts of data. So I think that is the real advantage of the WTA. And as far as using it for like diagnostics, we aren't at that point yet, but a lot of times the clients will come to us and ask, I have a drug that works, but we're not sure why it works, and we will certainly recommend the WTA or the whole transcriptome atlas at that point, because you can really look at several different signaling pathways on a whole genome level.
Well, great. A big thank you to Carlee, and thank you to our audience for joining us today and for their interesting questions. We will address any questions we did not have time for today and those submitted during our on-demand period via the contact information you provided at the time of registration. This session webcast will be available on demand after the event. That's all for this session. Thank you for joining us, and we hope you've enjoyed the presentation. Bye for now.